And the title of her talk today is 2 billion years of sheltering antelopes from telomere protection to cancer prevention. So just one precision for the audience before we get started. So you can ask questions using the Q&A or the chat. So Titia, the stage is yours, and we are very much looking forward to your talk.
Thanks so much, Aurelia, and thank you for inviting me and for your patience, because it took a year, I think, to show up. So I am, let's see whether I can get this to work. I'm going to talk about our work on Shelterin and T-loops, and in the second half of the talk, I will discuss our views on how telomere protection relates to cancer prevention. So the title of the talk is a little tongue-in-cheek, of course, but there is some truth to this. We do think that probably the Shelterin and T-loop system of telomere protection is very old, and may well go back 2 billion years to the origin of eukaryotes, which as you know, eukaryotes originated from an event where an archaea ate an alpha proteobacteria, which became a mitochondrion. And at this point, many things changed. Importantly for this talk, the nucleus was formed with a nuclear envelope, nuclear pores surrounding the genetic material. And it's reasonable to assume that at that point, the chromosomes were already linear. So even at that point, there was a problem with the linear chromosomes that needed to be protected from the DNA damage response. And I will argue that those telomeres and those linear chromosomes may well have already contained Shelterin and formed T-loops. So let's dig in a little bit into the telomere protection problem. So as you know, cells have vigilant ways of detecting double-stranded breaks and other lesions in the genome, which activate two main kinases, the ATM and ATR kinases, that can guide cell cycle rest, senescence, mitosis, and also modulate the types of DNA repair that take place at these breaks. And none of these outcomes would be appropriate if cells would take note of their natural chromosome ends, which of course resemble DNA breaks structurally very much. So in our cells, the telomeric DNA that is important to prevent this DNA damage response is composed of long arrays of TTA-GAG repeats and ends in, and this is very important for telomere protection, along 3' overhang of single-stranded G-rich repeats. This DNA itself does not solve the end protection problem, but it helps to recruit to the telomere and the Shelterin complex that Aurelia mentioned that binds very specifically to the telomeric DNA, and it's this complex that represses the DNA damage response. So this complex Shelterin is present all along the telomeric DNA, not just at the end. It binds the double-stranded telomeric DNA, and this is required for it to arrive at telomeres. Its single-stranded DNA binding activity is not sufficient for telomeric localization. And it binds to the double-stranded DNA through two proteins that are paralogs, TRF1 and TRF2, which have C-terminal MIP domains here, depicted as blocks that engage the telomeric DNA, and both here are shown as dimers, but I'll go into this in more detail. These proteins anchor the rest of Shelterin, bind RAP1, they bind TIN2, and TIN2 loads into the complex TPP1, and this protein interacts with POP1, a single-stranded DNA binding protein that was discovered by Tom Cech's lab that binds to the G-rich telomeric single-stranded DNA. Remarkably, this complex functions to protect telomeres from a whole host of different reactions, from activation of the ATM and ATR kinase, from both classical non-homologous end joining, homologous homology-directed repair, and also alternative end joining. And it represses DNA end processing, specifically 5' end resection. A major clue of how Shelterin solves the end protection problem was mentioned, and that's the formation of T-loops, a structure that we originally identified in collaboration with Jack Griffiths by simple EM analysis of telomeric DNA. But more recently, we've been able to develop techniques together with Zhao Zheng at Harvard to visualize telomeric DNA in spread chromatin by storm imaging, showing T-loops. So the T-loop is formed by TRF2. It's a structure in which this single-stranded telomeric overhang is strand invaded into double-stranded telomeric DNA. Could be any site along this long array of telomeric DNA. The strand invasion, of course, displaces a DNA strand, the D loop, but it's this strand invasion event that hides the chromosome end from the DNA damage response. It's not the loop per se that's important, but the insertion of the end, so that this site is occluded from both classical non-homologous end joining and ATM signaling. I think somebody needs to mute their microphone.
Okay.
Telomeres are threatened by classical non-homologous end joining, and when you delete TRF2, you can see that it's a big threat to telomeres. These are chromosomes from a normal mouse cell with telomeres at each end. If you delete TRF2 from mouse, and this was in old days, we did this by a Cre-mediated deletion of a flux TRF2 allele, you can see that all chromosomes become prone to undergo these fusion events where telomeres themselves are preserved, but they no longer are sufficient. The DNA is no longer sufficient to dissuade cells from thinking this is a double-stranded break, and cells execute the only reaction they can execute to take away this offending lesion, and that's classical non-homologous end joining. So as you know, classical non-homologous end joining is initiated by loading of the Q7080 heterodimer onto open DNA ends. It's a ring-shaped molecule that then recruits DNA PKCS, and DNA PKCS helps to recruit a number of processing and ligation factors that eventually seal the break. So we know that this reaction occurs upon deletion of TRF2, and we also know from work done by storm imaging by Uli Doksani that there is a loss of this T-loop structure when TRF2 is deleted. So the model then becomes very simple. The model becomes the following, that classical non-homologous end joining, by virtue of its need for a free DNA end, becomes blocked at telomeres where a T-loop is present because there is no free DNA end and Q7080 can't load. So this whole reaction is blocked at the initiation step. So this is also, the same principle also holds for how TRF2 represses the activation of the ATM kinase, which it does. This is a critical feature of TRF2 in Shelterin, is preventing the ATM kinase from becoming active. And the way this works is clear now that we understand from the work of many people in the ATM signaling field, not detailed here with references, that ATM relies on the MRN complex to become active. So Peter Hofner showed that the MRN complex attains a new conformation when it reaches the DNA end. It can zoom along the DNA, but when it reaches the DNA end, it forms a rod kind of conformation. This is an SMC-like protein that now has the ability to bind to the ATM kinase and converts, and this is the critical step, the inactive dimer of the ATM kinase into an active monomer by ways we don't understand, and don't take this cartoon very seriously. It cracks open this inactive dimer to form an active monomer that then can activate to lead to p53 upregulation, cell cycle rest, and it can modify components in the chromatin nearby the break. So again, the T-loop provides an architectural means of preventing this series of events from taking place, because in the T-loop structure, there is no topologically free DNA end. And so the MRN complex can never attain this critical conformation by which it can activate the ATM kinase. So this step never occurs at the telomere end unless the T-loop opens up. So this is a main clue about how telomeres work. By having a protein TRF2 that can form a T-loop that then blocks the ATM kinase signaling pathway in classical non-homologous adjoining pathway at the initiation step by preventing the critical initiating factors from gaining access to the telomere end. But there's, of course, a big question here that needs to be addressed, and that is, yes, we know from a number of experiments in my lab and other people's work that TRF2 is required for T-loop formation, and that no other Shelterin subunit is needed. And we also showed that the RAD51 recombinase is not required. It's an enzyme that could potentially make this kind of structure. So the question then is, how does TRF2, this fairly simple DNA binding protein, form T-loops? And we've worked on this quite extensively, and I'm going to summarize what we think is going on. We think that although I draw TRF2 everywhere in my schematics as a dimer, it actually has the ability to form a tetramer. It has additional dimerization interfaces that allow tetramerization. So the dimer is formed through this very large domain that is called the TRFH domain, by which TRF2 can interact with itself. This is a homotypic interaction that TRF1 also has. But we find that there is an additional interaction that allows two TRF2 dimers to come together when TRF2 is binding to DNA. And once you have a protein that binds to TRF2 DNA as a tetramer, containing now 4 DNA binding sites, you could imagine that this protein induces a loop that enforces torsional stress in the DNA. Because of the rigidity of DNA, when you force it to form a loop, there's torsional stress, and this may lead to opening of the helix that relieves that stress. And once you've opened the helix, you could allow the telomere terminus, which is somewhere in the vicinity of where TRF2 is binding. It's, of course, all over, because the telomere is not a linear thing, but a blob. This 3' overhang could now strand invade. And if you do that, you have a T-loop. So another way of drawing this is to think about TRF2 making these bubbles all over the place. And once the telomere terminus finds itself near a bubble, its strand invades. And now you have a T-loop, and this may actually be an unstable setting, a situation where the T-loop can open up frequently, but then that telomere terminus will find another bubble to reform the T-loop. So we came to this model from extensive biochemical analysis of TRF2, and it would take me an hour to go through that. So I'm going to shortcut it. It's all in a bioarchives preprint, and shortcut it and tell you how we ultimately provided substantial in vivo proof for this model. And that's based on manipulating TRF1. So TRF1 is very much like TRF2. I draw it as a dimer. Um, uh, it is not. It is a dimer, unlike TRF2. But unlike TRF2, it doesn't protect telomeres. It promotes replication of telomeric DNA, but it does not form T-loops in vitro or in vivo, and it does not repress the ATM kinase or classical non-homologous joining. So we thought we might test this model by simply converting TRF1 into a tetramer, because then it should start behaving like TRF2 in vivo. So, and that's not that hard to do. Because we thought we could fuse an inducible dimerization domain, such as the FKBP domain that can be induced to dimerize with the addition of AP1903 to the TRF1 dimer. It has its TRFH domain, so it's a dimer. And then induce the tetramer, and the inducible tetramer, we hoped, would behave like the tetrameric TRF2 and form T-loops. Now, these kind of experiments, as you well know, always look great on paper and in grant proposals, but they basically never work. But in this case, it did work. So what I'm showing you here are cells that lack TRF2 and express the tetrameric, the inducible tetrameric TRF1, and we're testing whether it forms T-loops by using olidoxanase method to spread telomeric DNA. So in cells lacking TRF2, there are no T-loops. When we reintroduce TRF2 into these cells, of course, we get T-loops. When we reintroduce TRF1, there are no T-loops, as expected. But when we introduce TRF1 with the tetrameric module on it and induce it, T-loops are formed remarkably. And this T-loop formation is demonstrably dependent on the ability of TRF1 to form a dimer, because if we delete this TRFH domain, then even with this dimerization domain, it does not work to form T-loops. So consistent with the ability of TRF1 to form T-loops, we see repression of ATM signaling. This is a readout, is the number of 53 BP1 foci at telomeres, a reliable readout for the activation of ATM. So if we delete TRF2 from cells, we get a lot of 53 BP1 at telomeres. It's rescued by TRF2, consistent with T-loop formation, not by TRF1. There are no T-loops there. And if we add this FKBP fusion protein, it itself cannot repress ATM, but with the addition of the dimerization a small molecule, it does. And this, as I said, is dependent on the TRFH domain of TRF2, and similarly, there is a repression of classical non-homologous injury. So this now suggests that this model holds TRF2, forms T-loops, and it's the T-loops in a TRF2 independent way that repress ATM signaling in classical non-homologous joining. And a recent paper from Jérôme Dujardin came to the same conclusion in a very different context. So this is a major solution to the end protection problem, but there is a second problem. To form a T-loop, you need a single-stranded telomere overhang, and this is also a great substrate for telomerase. So you need to do something about protecting this overhang from the other kinase, and that's the ATR kinase. And ATR signaling, as many of you know, is initiated by the single-stranded DNA binding protein, RPA, binding to single-stranded DNA. And through a number of other steps, this then indirectly leads to ATR signaling. This is the initiator. This is the sensor in the ATR pathway. And again, just like the sensors in ATM signaling and classical non-homologous end joining are prevented from binding at the telomere by TRF2 forming T-loops, the ATR signaling pathway is thwarted at the level of this sensor. And that's the job of POT1, which binds to single-stranded DNA. And by virtue of its tethering to Shelterin, is much better at binding to this single-stranded DNA than RPA is, even though it has the same affinity because it's locally tethered. It's very good in preventing RPA from binding. In addition, POT1 has a very special little thing here, which is called the pothole in its N-terminal OB fold. This lovely work from J.K. Kumar's lab, where POT1 binds over this 5' end, which is also essential for ATR activation. So I just want to return to my statement in the beginning that this is a very old system, that in my view, it's likely that the linear chromosomes in LICA already had telomere sheltering and T-loops. And I base that on our extensive analysis of telomeric proteins that have been worked up by other labs in other organisms. This was primarily a project that happened during the pandemic when we couldn't do anything but use our computers. And we dug back and looked at the conservation and the evolution of telomeric proteins. And so this is our Shelterin with its set of proteins. And this is specific for vertebrates. If you go back to Metazoa, it's pretty much the same, but the TRF1 is absent. So TRF2 was there, but TRF1 is a vertebrate invention. And then if you go way back to flagellates, trypanosomes worked on by Bibo Lee extensively. This is quite interesting because they have a TRF that is very much like TRF2. They have a TIN2 and they have RAF1. So they have a considerable number of these core proteins of Shelterin. And I think it's very likely that they also have POT1-like proteins because they're found in ciliates. This is how Tom Check originally found POT1 based on homology to a ciliate protein. So I think it's likely that all these players were there early on. In addition, if you look at the presence of T-loops, they're there, of course, in vertebrates, but they're also there in flagellates. Trypanosomes have T-loops, plants have T-loops, and ciliates in the micronucleus have been shown by David Prescott to have T-loops. So T-loops are also a very old feature. And if you look at the telomeric DNA, you can see that TTA-GG repeats by no means are a modern invention. At our telomeres, probably the first telomerase that ever evolved made this sequence and was somewhat changed in some organisms. So this leads me to think that the whole system is very old. I should say something about the fungi, which is interesting. Although Pombi has Shelterin that's very much like ours, Cerevisia has an entirely different complex. The only thing that's conserved is RAP1. It also has very different telomeric DNA, and it doesn't have, it is unlikely to have T-loops, although that was never really tested. So the question then is there nothing new at our telomeres? Are we working with an antique set of tools? I think there is news at our telomeres, and that in part is how Shelterin plays a central role in the recruitment and regulation of telomerase. So this is not work from our lab, but from a number of labs listed here. It showed that telomerase is recruited by a Shelterin component, TPP1, especially with this little nubbin binding to TERT, and this allows for the maintenance of the D strand. And in addition, we recently showed that Shelterin is required for the recruitment of the second machine that maintains telomeres, that is CST kinase that maintains the C strand. So Hiro Takai in my lab showed a little over a year ago that maintaining the G strand is not sufficient for maintaining telomeres. You also need an enzyme separate from DNA replication to maintain the C strand. Lovely collaboration with Joe Yields at the LMB in Cambridge showed that without this fill-in reaction by CST alpha primase, you cannot maintain the structure at the end. You can't fully copy the end of the telomere. And Sarah Takai, together with Hiro, showed that the way this machine is recruited onto telomeres is through an interaction with POT1 involving phosphorylation of POT1. So that's an interesting sort of elaboration that is specific for human cells. And in addition, Shelterin, of course, plays a critical role in regulating how telomerase act sequences onto the telomere. So it's long been known that telomerase preferentially adds telomeric DNA to the shortest telomere in a cell, not to the longest telomere. And the main model for how this works is the idea that a long telomere contains more Shelterin than a short telomere. And within Shelterin, there are a number of proteins that enact an inhibition on telomerase. It's a cis-acting inhibition that somehow, in ways we don't fully understand, switches off telomerase at a long telomere. We don't know at which step this occurs, and we don't know the mechanism of the switch off, but we do know the players. It's at least TIRF1, TIN2, and POT1 that are involved in these reactions. Now, this is very old work. As you can see, this goes back to ancient history when Bas van Steensel, you know, he was well known in other contexts, was a postdoc in my lab in the 90s. And at that time, we did not know that whether what we were measuring in tissue culture cells, this is what this is based on, telomerase positive tissue culture cell, actually holds in human physiology. And that changed once mutations were found in patients that came to the clinic that had loss of function mutations in POT1 or TIN2. They still had one normal copy. They're heterozygotes, these patients, and it turned out that these patients have very long telomeres. So this tells you that in the human setting, indeed, POT1 and TIN2 act to limit telomerase ability to elongate the telomeres. So the question is that you may have in mind is, why do these people come to the clinic? Well, they don't come to the clinic because they're worried about their long life or fewer wrinkles. There's very little evidence from these patients, in fact, that long telomeres help you age less. But what is very clear from these patients is that long telomeres lead to cancer. These patients come to the clinic because they're in families that are cancer prone. And some of the families that we studied show this, and it's quite dramatic. I point you to this patient who had a TIM2 mutation, one loss, one null allele, one normal allele, and she had 5 cancers, 5 independent primary tumors before the age of 70. So it's a heavy tumor burden. And anybody who lives to 70 without getting this many cancers can thank their telomeres in part for protecting them from cancer development. So this told us 2 things. Telomere length is kept in check by Shelterin early during development and probably in the germline and in stem cells as well. It also helps to allow for the telomere tumor suppressor pathway to enact itself. So the idea about the telomere tumor suppressor pathway is that telomerase, which is on early on in development, is silenced in somatic cells, including somatic cells that attain a mutation that allowed them to proliferate more than is appropriate for that particular tissue. And the cells, once telomerase is silenced, will undergo telomere shortening because of the end replication problem. And after a given number of cell division, estimated to be 50 or so, there will be telomere dysfunction because there are a few telomeres that are so short that they're no longer protected. There presumably is insufficient Shelterin at the telomeres to be protected. This then leads to a DNA damage response that enforces apoptosis and senescence. And this, of course, is a tumor suppressor feature and can explain why people with long telomeres have a delayed Hayflick limit, which is what this point in the growth of cells is called, where the culture stops dividing. So to illustrate the thought here, if you're born with long telomeres, because Shelterin is deficient in its ability to inhibit, to control telomerase, it will take many more cell divisions to reach the Hayflick limit, to reach the point where there's sufficient unprotected telomeres to induce the cell cycle arrest. And during this delayed Hayflick limit, because of it, there will be divisions that will allow the accrual of additional mutations that predispose to, that increase the cancer risk. So because of this new realization that, as we had anticipated for about 30 years, telomere shortening indeed is a very powerful tumor suppressor pathway. Alex Stewart in lab returned to the question of what happens in the Hayflick limit. This question had been addressed by John Sedivi and by Steve Jackson's lab, who both had shown that indeed it's a DNA damage response. It's signaling of the ATM and or ATR kinase that leads to apoptosis and senescence. But what Alex wanted to know is more precisely, is it ATM or is it ATR? Because you can imagine that a short telomere that has a hard time recruiting Shelterin because there's so little binding DNA may either lack sufficient POP1 to repress ATR signaling or may lack sufficient TRF2 to repress ATM signaling, or both. And so the way Alex addressed this is by using simple inhibitor chemical inhibition of ATM and ATR and ask, is the Hayflick limit, the induction of senescence, dependent on ATM and ATR? And the data was published a few weeks ago, so I'm not going to show it all, but it was very clear that from his experiment that it's ATM that is responsible for the Hayflick limit. ATM inhibition extends the lifespan of primary human cells, senescent cells, enter the cell cycle when you treat them with ATM inhibitors. So it's partially reversible. You can get them back in. When you take away ATM signaling, none of this is seen with ATR. And importantly, TRF2 overexpression, loading up more TRF2 on those short telomeres, extends the lifespan of primary human cells. And note that I say here at both 3 and 20% oxygen, and that is important. Sorry. And I'll come back to that in a minute. So, simple interpretation here is that the few unprotected telomeres that have insufficient TRF2 to repress ATM signaling simply cannot form a T loop to prevent the activation of the ATM kinase. But there are other possibilities that we're testing right now. And we also don't know what the length is of these critically short telomeres. And we're trying to address that as well. Okay, the reason why I noted the oxygen levels in 2 slides back is that it's long been known that cells have the ability to execute more cell divisions when they're grown at physiological oxygen, 1 to 5%. Now, physiological oxygen is quite different from what we call normoxia. There's nothing normal about normoxia, which is around 20%. No cell in our body or any mammal experiences 20% oxygen unless you cut open the tissue, and perhaps cells in the upper trachea experience this. But this is the physiological oxygen level. So rather than saying that proliferation is enhanced and senescence is delayed at lower oxygen, you could say it's accelerated. Senescence is accelerated at 20% oxygen. So you can see here from this growth curve. So Alex wanted to know what's going on, and others had already provided compelling evidence against the idea that at higher oxygen, the telomeres simply erode faster. This would, of course, a higher erosion rate would, of course, explain why you would senesce earlier at 20% oxygen. But a number of labs had shown that that really isn't the case. And we also showed this here by universal single telomere length analysis, or STELLA, which is a PCR-based analysis of telomere length, where each band represents a telomere. And you do 3 PCRs per sample, and then you can measure the length of each band really carefully. So you can see there's high variability in telomere length, and each PCR grabs a different subset of telomeres. You don't see 100 telomeres per lane, just a few. And you can see here that because these two PD points are just before these two cultures senesce, it's a good point to compare them. And you can see that at 3% oxygen, as these cells are growing, the telomeres are not longer than at 20% oxygen. If anything, they look a bit shorter on average. And what is particularly striking is the larger number of very short telomeres. So we, without any particular reason, put a line at 1.8 and counted the bands below versus the bands above. And you can see if you plot that the a higher number of shorter telomeres at 3% oxygen. So this tells you that these very short telomeres are better tolerated at 3% oxygen because they're not allowed to exist at 20% oxygen. And this led Alex to ask whether perhaps cells in general are less responsive to DNA breaks at low oxygen. So he did a very simple experiment, simply treated his cells with zeosin at 20% oxygen and 3% oxygen. And visually, it's very easy to see that there are fewer gamma HCOX foci induced by zeosin. There's a readout for ATM activation. Many fewer foci at 3% and 20%. And all the controls worked out fine. Zeosin doesn't affect double-stranded break. Oxygen doesn't affect double-stranded break induction by zeosin. And there's a similar result with etoposide. We also see this result when we induce tumor damage in RPE cells, and there is no effect of oxygen on the ATR response induced by hydroxyurea. If you quantify this, it's a very convincing drop into primary human cell lines at 3% oxygen. Just don't respond to zeosin as much as at 20%. So this raises the question of why, how ATM is attenuated at low oxygen. And we, of course, asked very obvious questions about the ATM kinase pathway. Is the level of ATM down at low oxygen? Is there less MR11, less RAD50, less MBS1, et cetera, et cetera? None of that turned out to be true. So we then turned to thinking about the work of Tanya Paul, who had shown in original science paper that there is a second way of activating ATM that involves formation of crosslinks between the monomers in the ATM dimer. So recall the ATM dimer normally is inactive, but if you crosslink this dimer, apparently this is cysteine bridges cross-linking in dimer, you can activate this kinase and it becomes a very different kind of kinase. It can no longer be activated at DNA ends. Obviously, it cannot be dissociated. And this kinase functions to counteract factors, to counteract the effects of ROS. And it's in fact ROS that induces the cross-linked situation of the ATM dimer. So there's a wonderful way in which the ATM dimer can take care of problems that arise when ROS occurs, because ROS cross-links the dimer, activates it, and now cells can, through phosphorylation of factors, better tolerate ROS. So is this involved in our attenuation? Well, for that to be true, for the idea being that you have so much dimer cross-linked that there is very little ATM kinase left over to be activated at double-stranded breaks, most of them will be blocked. For that to be true, you would have to have higher ROS at low oxygen. And this was disconcerting to me. Why would there be higher ROS at low oxygen? I always thought naively that there should be lower ROS at low oxygen. But Alex reads the literature and convinced me that this is not a crazy idea, that it had been published, that there is higher ROS at low oxygen, and he reproduced this data using two readouts, superoxide and ROS. You can see that in RP cells at low oxygen, there's much more ROS here quantified. So there is more ROS at low oxygen, which could potentially explain why there is less ATM activation at double-stranded breaks. And for this to hold, we argued that we should be able to revert this situation at low oxygen by undoing the crosslinks with either N-acetylcysteine or TSAP. And that's true. So you take RPE cells at 3%, you treat them with zeosin, you get very little ATM response, you add NAC, now you get a much higher ATM response. Alex went on to look at the presence of this dimer in cells at low oxygen. The non-covalent ATM dimer falls apart during isolation. It doesn't stick together very well, but the cross-linked dimers stay intact. And so if you do a mild extraction and then you immunoblot from a native non-denaturing gel, you can see the dimer, and it's less at 20% and reduced by NAC. In addition, in collaboration with Katia Vinogradova, we identified some of the cysteines that are involved in this cross-linking by using her wonderful chemical proteomics platform. And the cysteines are shown here as a relative reactivity at 3% versus 20% oxygen. So this one is reduced in reactivity. That is, it's less exposed, therefore more involved in cysteine bridges. So there are a number of these. We by no means have identified all of them, but one was very noteworthy to us because cysteine 2991 had already been implicated in the activation of the ATM kinase by cross-linking by Tanya Paul. And so we asked, is this cysteine responsible for needed for the effect? Cells provided by Tanya Paul, in which this version of ATM or the wild type was overexpressed. And we do the same experiment again. We look at H3X foci in cells that have been treated with leucin at low oxygen. And so again, very few foci. But when we use that mutant, now we have a much better response. So this is entirely consistent with this idea of cysteine cross-linking at low oxygen where there is higher ROS, creating a pool of ATM that can't be activated at critically short telomeres or double-stranded breaks. So I'm going to summarize now. I'm out of time. So what Alex showed is that replicative senescence is an ATM-dependent process in which telomere shortening creates a critically short telomere that has insufficient TRF2. And at that telomere, it's the ATM kinase that becomes active and induces replicative senescence through its well-known monomerization. And we can take senescent cells and incubate them with the ATM inhibitor and kick them back into cycle. Alex also showed that the long-known longer lifespan at low oxygen is due to a higher level of ROS. ROS-induced cross-linked ATM dimers, which can't respond to critically short telomeres, take away a lot of the pool so that there is an attenuated response to double-stranded breaks and to short telomeres that lead to this longer lifespan that is normal replicative senescence. Whereas at 20% oxygen, we basically are working under hyperactivated ATM conditions, increased pool of ATM that is not cross-linked, that can respond to double-stranded breaks. And so you get that accelerated senescence. So in summary, I told you that Shelterin and T-loops are likely to be a very ancient system, perhaps dating back to the origin of eukaryotes. I told you how Shelterin controls telomere maintenance through its recruitment of both the CST Pol alpha primase complex and telomerase, and it controls telomere length. And the control of telomere length is critical for the execution of a proper tumor suppressor pathway, because it's the telomere shortening and induction of growth arrest that protects from cancer in a very potent way. And if you're born with telomeres that are not of the appropriate length, where Shelterin has failed to control telomere length, this pathway is delayed, and this gives rise to highly tumor-prone syndrome. And finally, I showed you that the critical induction of the growth arrest that takes place in this tumor suppressor pathway is due to the activation of ATM, which is affected by oxygen levels through an effect that involves ROS. So I will end with acknowledging the people in my lab, too many contributions to Nodal. I talked about Alex's work. He's now at Harvard doing a postdoc. Here are the current people in the lab, and I need to acknowledge my funding, without which we wouldn't exist. Thanks so much.
Thank you very much, Titia, for this very inspiring lecture. It was really an amazing overview of your work. Just to remind the audience, so please ask your questions using the chat, or raise your hands and we will unmute your microphones. You can just ask the first one while people type in their questions. So about the patients with the POT1 mutations, so do you see any specific genomic features in the tumors that develop in these patients as compared to patients who have who don't have the POT1 germline mutation?
I should stress that the POT1 mutations aren't my work. We worked on PIN2 mutations. The POT1 mutations were originally identified in 2014 by two groups in melanoma patients. These families are quite rare, and I don't think anybody has looked at specific hallmarks. Genomic hallmarks. I wouldn't expect any, because these mutations do not change the protection of telomeres. There still is a wild-type copy of the protein there, which is sufficient for protection of telomeres. So I wouldn't expect any kind of additional genomic instability in these patients.
Okay, so yeah, there is one question from Francisco in the chat. So very interesting talk. Thank you. What outcome would you expect in hypoxic conditions? Would you expect an even longer lifespan?
So in hypoxic conditions, cells are very unhappy in culture. So their lifespan is a lot shorter. So there is the question, so 1% is not hypoxic, but there's, of course, as you go down, right, there might be a point, maybe 0.95%, where you get the longest lifespan. Because the lifespan at 1% is actually longer than at 5%. So I don't know where that cutoff is, but if you really become hypoxic in tissue culture, the cells are very unhappy.
One question from Flora. So since the telomeres can be elongated by telomerase, but there is also another mechanism called lengthening of telomeres, would this cause any difference in the chromosomal instability?
I don't think I understand that question.
So whether there is a difference in the chromosomal instability, whether telomerase is activated or ALT is activated.
So when telomerase is activated without any other change in the cell, as Jerry Shea showed originally in the 90s, there is no genome instability. It's completely inert situation. No cells remain diploid, no fusions, nothing. When you look at old cells, they have massive chromosomal instability. So old in cancer and in vitro is inherently driving some form of chromosome instability.
Thanks. There is one more question from Akana Krishnamurthy. So great talk. Does tetrameric TRF1 still support replication of telomeric DNA?
We did not test that in the strictest way, which would be to express that tetrameric form of TRF1 in TRF1 null cell. But it's overexpressed. TRF1 FKBP is overexpressed about tenfold in our setting. And if it was non-functional, we would have expected to see evidence of replication stress at telomeres, ATR signaling, which we don't, and fragile telomeres, which we don't. We know that the repression of replication problems at telomeres is very much dependent on having the full load of TRF1 at the telomeres. So, when most of the protein at the telomeres is the tetrameric form and only 10% or so is the wild type form, we would have expected to see a phenotype if it was deficient, but we haven't strictly tested that. I should also remind you that the way TRF1 protects against replication stress is purely through protein-protein interaction. It binds TF2H, it binds BLM, it binds TIM2, and therefore brings POT1 to telomeres. And these protein-protein interactions are critical to help the replication of telomeres. And all of those features are preserved in the tetrameric form. But yes, we haven't tested it strictly.
Thank you. I see one more question from Amanda Meni. Is there a relationship of the nucleosomes to Shelterin and the ability to form T-loops?
There are nucleosomes at telomeres. I never draw them in my schematics because it's just too hard. And they may have special features. We're not sure about that. Shelterin doesn't seem to be bothered in its binding to telomeric DNA by the presence of nucleosomes. The nucleosomes don't change when you delete Shelterin, so it seems like a neutral aspect of telomeres. And we know that Shelterin can form T-loops in the presence of nucleosomes, and Woodcock a long time showed in chicken telomeres that T-loops have nucleosomes on them all through the loop and the tail. There are nucleosomes there. So I don't think there is a story there, but I can't strictly exclude it.
Thank you. I cannot see any more questions in the chat. So thank you very much again, Titia, for this fantastic
You're
seminar.
very welcome.
It was a great pleasure to have you with us today.
Okay.
So our next lecture will be on the 6th of March. I think Florian has the schedule in the chat. And so thank you very much again, and thank you all for joining, and enjoy the rest of the day.